So, hello everyone. Today we are going to discuss the next lecture on cell nutrients. So, in this module, first we started with fundamentals of bioprocessing. So, there substrate is converted to product using biological agent. Then further in the last lecture we discussed the metabolism, both catabolism as well as the anabolism, that how a substrate is converted to different intermediates and different product. Then today we are going to discuss nutrients, because once you have got your desired microbe, which was also discussed in the last lecture, that isolation of potential strains, once you have isolated, further you have understood its metabolic pathway. Now, next comes that media or the substrate. So, today we are going to discuss about the nutrients which is required for their growth, maintenance as well as for the product formation. So, as we know this first law of thermodynamics, that energy is conserved, similarly mass is also conserved. So, considering this mass balance principle, you have to provide the substrate, and at the end it will either go in terms of increase in the biomass or it will go in terms of products. So, at the end total mass will remain constant. So, as you know, the cell is having certain chemical formula, and in addition it has certain internal composition, and that is very different from its external environment. And in order to grow, a cell must import useful compound and retain essential molecule. And how it is done? It is done through the selectivity. For example, you can think of semi permeable membrane, it is selective for some elements or some ions. Further, maintaining internal condition away from the equilibrium requires continuous energy expenditure. So, it depends whether this transport is active transport or passive transport. Further, this membrane plays a crucial role in homeostasis and selective transport. For example, you can see sucrose, sodium, potassium, glucose, tryptophan, glycerol, urea, ammonia, water. So, this is the increasing permeability for these molecules. Now, nutrients we can classify as micro as well as macronutrients. So, cell requires carbon, nitrogen, oxygen, hydrogen, sulfur, phosphorus, magnesium and potassium as macronutrient in the large quantity. In addition, they also require the micronutrients in minor quantity like trace element, vitamins and hormones. Typically, if you see, a cell contains about 80% of water, and if I see the dry cell composition, it is mostly proteins, 10 to 20% nucleic acid and 5 to 15% is lipid. However, virus may contain up to 50% of the nucleic acid, because it only contains nucleic acid and protein cover. Further, the cell composition vary with different cell types. what is the age of a particular cell and depending on the availability of nutrients. In addition, these environmental conditions like the pH, temperature, oxygen supply or PM, it also affect the metabolic product formation. So, you can see it can be classified as macronutrients and nutrients means concentration less than 10 to the power minus 4 mole, and these are the macronutrients and these are micronutrients. Now, you can see this typical composition: protein, nucleic acid and lipid. In the virus, it does not contain lipids. So, it is mostly protein and nucleic acid, but in bacteria if you see majority is protein, it can be up to 70%. Further in the yeast also it is 40 to 50% is protein. In filamentous fungi it contains 10 to 25% proteins, nucleic acid and lipids. And if I see this algae, it contains protein. The figure in the bracket is commonly found below, typically 50%, nucleic acid 3% and lipid about 10%. And typical population you just see all are in the range of 10 to the power 8 and above, and this is a typical dry weight of this culture. So, it is less for the virus because of its smaller size. Further, when you go for this filamentous fungi, it has a large structure, larger in size, so its dry weight is also higher, and this is for bacteria 0.02 to 2.9, and similar range is for the yeast 1 to 5 gram per 100 ml. Now, we will be discussing few selected macronutrients. So, first we will be starting with the carbon. This is very much essential for forming skeleton and backbone of organic macromolecules, and if you are going to form the hydrocarbon, this hydrogen and oxygens are also required here. In addition, electrons are needed for energy production via electron transport chain. So, we discussed the electron transport chain in the last lecture of metabolism. Further, they require reducing molecule in biosynthesis like NADH, FADH2. So, you can see based on the carbon source, energy source and electron source, we can classify them autotroph if carbon source is carbon dioxide, heterotroph if carbon source is chemicals. Energy source phototroph if it is sunlight, chemotroph if it is chemicals. Electron source lithotroph means if it is inorganic molecule, organotroph if it is using organic molecule as electron donors. So, first as I told we will discuss with carbon. So, common carbon source is molasses, starch, corn syrup, waste sulphite liquor. So, typically we see if there is a waste material which are rich in the carbon, so they are mostly exploited in the bioprocessing. In fact, nowadays this cellulose or cellulosic biomass is also being explored. So, when we start this laboratory scale experiment, we use glucose, sucrose, fructose, glycerol. So, these are easily available and which have well defined composition. Sometimes few strains also require methanol, ethanol or methane as a carbon source. So, the bioprocess can be aerobic or anaerobic. In case of aerobic, 50% of the carbon is used in the cell biomass. So, typically in the aerobic condition there is a more production of biomass, while in the anaerobic condition only 30% used in the biomass and most is converted into product. Like if I say in case of anaerobic digestion, the product is methane gas. Next is nitrogen source. This is needed for the, as I told, amino acid have nitrogenous compound, nucleic acid have nitrogenous compound, carbohydrates may also have nitrogen requirement, lipids and enzyme cofactors. Now, for nitrogen source one can have inorganic and organic nitrogen. Inorganic nitrogen, ammonia, ammonium salt like ammonium chloride, ammonium sulphate, ammonium nitrate and even urea. Organic sources, protein, peptides, yeast extract, peptone. So, this yeast extract and peptone are quite common. In addition, there is another route of nitrogen fixation, particularly this azotobacter and this cyanobacteria converts atmospheric nitrogen into ammonium. Oxygen, particularly if you are talking of aerobic metabolism as a terminal electron acceptor, typically it constitutes 20% of dry cell weight and it is supplied by the process of aeration. It may be surface aeration or it may be spurging. Hydrogen, it constitutes 8% of dry cell weight, basically derived from the carbon source, hydrocarbons. There are few methanogens, they use hydrogen as their energy source. Phosphorus, it is about 3% of dry cell weight. This is found in nucleic acid and cell wall. For the supply of phosphorus, inorganic phosphate salt KH2PO4, K2HPO4 are most commonly used. It also participates in the metabolic regulation. For example, phosphate less than 1 millimolar in media promotes secondary metabolites production. Sulfur, this is 1% of cell dry weight and this is present in proteins with sulfur containing amino acids and the source is sulfate ions like ammonium sulfate. Potassium, it works as a cofactor for enzyme. Cell actively uptake potassium and magnesium while excluding sodium and calcium. So, this is a ion transport. And common salt for potassium is K2HPO4, KH2PO4, K3PO4. Magnesium, this is again required as a cofactor for enzymes and it is essential for ribosome function, cell wall and membranes. Common salt is magnesium sulfate, magnesium chloride. So, you can see this 8 macronutrient element: carbon, nitrogen, hydrogen, oxygen, sulfur, phosphorus, potassium and magnesium, and typically they are required concentration in mole per liter. This is greater than 10 to the power minus 2, nitrogen is required in the less amount, further sulfur is further less amount, phosphorus, potassium and magnesium in the similar range 10 to the power minus 3 to 10 to the power minus 4 mole per liter. And these roles we have already discussed, like carbon, it constitutes organic cellular material, nitrogen, protein and nucleic acid and so on so forth. So, this we have already discussed in the last slide. Now, major nutrient type of microorganisms. So, as we have discussed, it can be photolithotrophy, photoheterotroph, chemolithotroph, chemoheterotroph, and chemoorganoheterotroph. So, here the carbon source is carbon dioxide, energy source is light. For example, the cyanobacteria, they are photolithoautotroph. Similarly, photoorganoheterotroph, carbon source is organic carbon. Photo means energy source is light. Heterotroph, this requires the organo means organic electron donor. So, this is purple non-sulfur bacteria, green non-sulfur bacteria. Next is this chemolithoautotroph. So, as the name suggests, autotroph means carbon source is carbon dioxide, chemo inorganic chemical, lithotroph is an energy source inorganic chemicals, and inorganic electron donor. This is sulfur oxidizing, hydrogen oxidizing, methanogens, nitrifying, iron oxidizing bacteria are a few examples. Chemolithoheterotroph. Chemolitho means inorganic chemical for energy. Chemo means this organic carbon as a carbon source, and few example is some sulfur oxidizing bacteria. Next is chemoorganoheterotroph, means this is carbon source is the organic carbon and energy source is organic chemicals in place of inorganic chemicals. This is common in most non-photosynthetic microbes including pathogens, fungi. Now, next comes micronutrients. Micronutrients, you can say it may be trace element, growth factor or chelating agents. So, what is micronutrients? This require in the concentration less than 10 to the power minus 4 mole. The importance of trace elements in microbial nutrient, these are essential for microbial growth, and its deficiency prolongs the lag phase and decreases the growth rate. Most widely needed trace elements are iron, zinc and manganese. Iron also works as a cofactor in ferredoxin and cytochrome. It regulates the riboflavin and penicillin production. Zinc, it regulates the, for example, it regulates the penicillin fermentation. Manganese, it regulates the secondary and primary metabolite excretion. Now, trace elements, so we have discussed trace elements, growth factor and chelating factor. The trace elements needed under specific growth conditions. For example, copper, cobalt, molybdenum, calcium, sodium, chloride, nickel and selenium. So, copper found in the respiratory chain enzymes and its deficiency stimulate penicillin and citric acid production. So, it is not necessary, it is required, even its deficiency stimulates penicillin and citric acid production. Cobalt, it is required by propionic bacteria and methanogens. Molybdenum, it is a cofactor for nitrate reductase and nitrogenase. This is needed for nitrate and nitrogen metabolism. Calcium, it is cofactor for amylases and proteases and found in bacterial spores and cell wall. Sodium, this is needed in trace amount by methanogens and it is essential for iron transport in eukaryotic cells. Chloride, it is required by halobacteria and marine microbes. Nickel, this is cofactor for certain methanogens, and selenium required for formate metabolism in some organisms. Now, third is the rarely required trace elements like aluminum, silica, even chromium, titania, they are needed in extremely low concentration, less than 10 to the power minus 6 mole, because they are toxic at higher concentrations. In addition, chelating agents, they prevent ion precipitation. Some chelating agents are magnesium, iron, phosphate. So, chelating agents form soluble metal complex using carboxyl, amine and mercapto groups. For example, this EDTA, this is most widely used chelating agent. It may cause cell wall disintegration by removing calcium, magnesium and zinc. Citric acid, this metabolizable by some bacteria. The typical concentration of a chelating agent is around 1 millimolar. Next, growth factor. So, there are three major classes of growth factor: amino acids, purines and pyrimidines and vitamins. Amino acids are needed for the protein synthesis and required at a concentration of 10 to the power 6 to 10 to the power minus 6 to 10 to the power minus 13 mole. Purines and pyrimidines are required for nucleic acid synthesis, as we have discussed in the last lecture of anabolism. And vitamins are a small organic molecule that usually make up all or part of enzyme cofactors, and they are needed in a small amount, 10 to the power minus 6 to 10 to the power minus 12 mole. Hormones, they regulate the metabolism in higher life forms, for example, insulin and in animal cells and cytokinins in plant cells. They stimulate growth and metabolite synthesis. So, these are the functions of some common vitamins in microorganisms. You can see biotin, cyanocobalamin, folic acid, lipoic acid, pantothenic acid, pyridoxine, niacin, riboflavin and thiamine. So, these are all common vitamins. Like biotin, function is carboxylation. B12 is needed for the molecular rearrangement. Similarly, if you see this thiamine, this is required for aldehyde group transfer. Riboflavin for the precursor of FAD and FMN. Niacin precursor for NAD and NADP. And these are the example of microbes which require these vitamins. For example, biotin is required by Leuconostoc species, Saccharomyces cerevisiae. Vitamin B12 is required by Lactobacillus species. Folic acid is required by Enterococcus species. Lipoic acid is required again by Lactobacillus species. In addition to bacteria, you can see F stand for fungi and P stands for protist. So, let us say niacin, this is required by bacteria, this is also required by the fungi and this is required by the protists. Thiamine again required by Bacillus species, Phycomyces fungal species and Archomonas protist. So, in this way we can classify these nutrients as micro and macronutrients. Now, after knowing that those nutrients should be present in the culture media, then only the microbes should be able to utilize them. So, culture media can further be classified based on the chemical composition, based on the physical nature and based on the function. So, based on the chemical composition, we say it is simple media, defined media or complex media. Based on the physical nature, you can say liquid media, semi-solid media, solid media or biphysic media. Based on the function, whether it is a supportive media, enriched media, we discussed enriched media yesterday in the lecture of microbial isolation, where after isolating we need to increase their population. So, we use enriched media, then selective media, differential media, indicator media, transport media and anaerobic media. So, you can classify based on the function, you can classify based on the physical nature, and you can classify based on the composition. So, physical nature, it can be liquid, solid, semi-solid. Chemical nature, it can be defined or the complex, and functional nature, it can be supportive, enriched or selective. Now, growth media, as I discussed in the last, it can be a defined media or the defined we are saying synthetic, or it can be a complex media. So, defined media, it contains a specific amount of pure chemical compounds with known composition. For example, media with glucose, ammonium sulfate, magnesium chloride and KH2PO4. So, it is a source of carbon, nitrogen, sulfur, phosphorus and magnesium. What is the advantage here? You can get more reproducible result, because all the metabolism will depend on the nutrients which is supplied. You can better control of the fermentation condition and easier and cheaper product recovery, because we know what are the compound in the substrate, whether they have been utilized or still remaining in the product. So, this advantage, it may require additional supplementation for the growth factor, trace elements, and typically it gives lower cell yields as compared to the complex media. The defined media, this like BG11. So, you can see all the components: sodium nitrate, potassium phosphate, magnesium sulfate, calcium chloride, citric acid, ferrous ammonium citrate, EDTA, sodium carbonate, trace metal solution, and this is final pH 7.5. Everything is defined. Similarly, one medium for the equally glucose, sodium hydrogen phosphate, potassium hydrogen phosphate, ammonium sulfate, magnesium sulfate, calcium chloride, iron sulfate, and final pH is this range. Next comes complex media. So, basically it is a natural compound with unknown chemical composition. For example, yeast extract, peptone, molasses, corn strip liquor. So, as it contains different micro and macronutrients, so they result in higher cell yield due to presence of growth factors, vitamins and trace elements, and they are often cheaper as compared to defined media. But it also has certain disadvantage, like less reproducible results, the more difficult to control fermentation condition, and as this unutilized media part will go along with the product, so recovery and purification becomes more complex here. So, here few example of complex media like nutrient broth, it is having peptone and beef extract. This triptych soy broth, it has tryptone, peptone, glucose, sodium chloride and dipotassium phosphate. Now, we will quickly discuss this mechanism of action of selective and differential media, because some media may be selective as some may be differential. Selective means they will only allow the growth of a desired population of microbes. Like for example, this blood agar, this is enriched and differential media. So, this will only support the growth of many bacteria. In addition, it will also help in the differentiation. But when we are having this eosine methylene blue agar, it is a selective and differential. So, this is eosine and methionine blue inhibit the gram positive bacteria. So, only gram negative bacteria will be able to grow. For example, MSC agar, this is again selective and differential, they inhibit the gram positive bacteria. This next is this mannitol salt agar, this is again selective and differential, it is selective for Staphylococci. So, pathogenic Staphylococci ferment mannitol, which is there producing acid and thus turns phenol red indicator yellow. So, for example, if you are plating or identifying Staphylococcus aureus, you can use this mannitol salt agar media. So, with this I will stop here. So, today we discussed the different nutrient requirement. It has been classified into this macro and micronutrients, and further we discussed the type of cell culture media. It can be based on its composition, defined and complex. It can be based on its physical nature like solid, liquid or semi-solid, or it can be based on the function like it is a selective media, enriched media. Thank you. I will stop here. Thank you.